My name is Roger Nisbet, and I'm going to give a presentation on one particular tool for modeling that is being increasingly used across a range of potential applications, so-called dynamic energy budget models. Specifically, what this presentation is going to focus on is the use of these so-called dynamic energy budget models as a tool for connecting data from different levels of biological organization. The material I want to be presented has been greatly influenced by collaboration with a large number of people, but I want to particularly thank those named in the front slide there, many of whom were participants in the so-called University of California Center for Environmental Implications of Nanotechnology, and also participants in two workshops at the National Institute for Mathematical and Biological Synthesis, from which much of the synthesis in these in this presentation derives. And the presentation, in fact, starts by stating what I think are a number of premises that are broadly accepted by people working in ecology. The first is perhaps the obvious one, that scientific method requires generating and testing hypotheses. Another is that ecological models should yield testable predictions. And I'm particularly interested in coherent ecological theory, which is perhaps distinctive from models in that theory is characterized by yielding many predictions from a small number of ideally rather general hypotheses. The relevance to this talk is that many fundamental ecological questions relate to the effects of environmental change on populations, communities, and ecosystems, sometimes referred to as different levels of biological or ecological organization. And it's my belief that understanding ecological effects of changing environments requires integrating information across levels. The challenge is, of course, that the predictions of this integrated theory has to be tested. A simple way of thinking about this is that there is this great divide between the type of information we obtain, especially from lab studies, and what we care about, which are typically processes at the level of populations, communities, and ecosystems. And this divide is reflected in the nature of the data that we get and the quantity of data we get. At the lowest levels of biological organization, deep suborganismal level, or at the level of unicellular or very simple organisms, there's huge amounts of data available. There are high throughput methods that get us more and more data. They're of biomedical relevance, therefore there's gobs of money going towards gathering these data. As you move up, and just imagine the middle pair of pictures in my slide, where you're looking at in the top picture a Daphnia, a small water flea. Information is a bit harder to get, but there's still huge numbers of study. When you reach the endpoints that are of societal importance, the top left is a mouse, the bottom left is a stream. The amount of effort that can be obtained is just a small number of, a relatively small number of data samples per year. And for me, a challenge for theorists is to use this information from organismal and suborganismal studies to prioritize, guide design, and interpret ecological studies, and specifically to inform ecological risk assessment, which implies prediction. And the tool that I'm going to describe is a tool known as dynamic energy budget modeling. Dynamic energy budget models describe the assimilation and the use of energy and elemental matter by living organisms. At the heart of dynamic energy budget modeling or theory is a model of a single organism. This model uses a set of differential equations. That's a set of equations that describe describe changes over time of quantities that relate to the growth, the reproduction, and some components of the mortality of an organism, and they relate these to its food intake and to set of environmental conditions. The definitive text defining dynamic energy budget theory is the first of the set at the bottom of the slide here, the book by the Dutch theoretical biologist Bas Kooijman. It's a pretty terrifying, impenetrable book, though it's wonderfully authoritative once you actually get into it. And for that reason, I've given a second, still pretty comprehensive overview, which is the second paper, which was by a group of authors led by Marco Yusup, and this was published early in 2017. At the bottom of the list of references, I've given a reference to some a URL for some lectures that I gave a few years back at the Center for Environmental Implications of Nanotechnology. If you want some details presented in a somewhat different way, that link is there, and I checked before starting to give this presentation that the link was still working, and it is. Dynamic energy budget models have the potential for connecting effects at different levels of organization, and I'll elaborate on the details there, because we can use them in so-called individual-based population models. And you can think of individual-based population models as being big computer simulations where you, in a sense, throw into the computer a bunch of organisms, every one of which is obeying the rules of dynamic energy budget theory, and then you just let it rip and you watch what happens, and that way, in a sense, you're predicting population dynamics. There are many flavors of dynamic energy budget models and even many variants on the version proposed by Bas Kooijman. I'm only going to describe in this lecture, and very, very briefly, one that is sometimes called his standard model. This model describes all the life stages of an animal, and it involves anything from up to 6 individual level state variables, depending what we want to predict, and anything from 2 to a dozen or even more parameters. And the next slide is a schematic that describes what this model, essentially how this model works. It's the standard kind of box and arrow slide that you've probably seen in other contexts, where arrows represent flows and boxes represent containers with stuff inside them. And this model characterizes the complexity of an organism in terms of a rather small number of variables, one of which is called structure, one of which is called reserve. And for this presentation, we don't need to worry about the definitions of these quantities. Essentially, food is assimilated by the animal, and the form in which it enters the animal is called reserve. You may find that a bit unfortunate, but that's just jargon. And then it's utilized for multiple purposes. It's utilized to make new structure. It's utilized to support development or increase of maturity. It's used to meet maintenance costs. And of particular importance within the model, there is a physiological feedback mechanism that regulates the proportion of the animal at any one, or the organism, but it's commonly an animal, at any one point that is at any particular time, so that to some extent the balance of the amount of material in the different compartments remains constant. The next slide shows how this can be relevant at higher level of organization, because all I did to go from the previous slide to this slide was put a little box around it and shaded what was behind it, a sort of bluey purple. And what that emphasizes is that what we're now describing is the whole organism. So we can now imagine lots of entities like what is on this slide interacting with each other and with the environment, and we have a route through to having some kind of ecosystem perspective still based on the dynamic energy budget model. So how do we do that? We first have to define a set of so-called state variables. State variables is dynamic modeling jargon. All it means is things that vary over time in response to the differential equation. I said there could be up to 6, and I've listed them there in the slide. And the first 4 come from the pictures you've just seen. The bottom 2 are ways of characterizing mortality and enhancement of mortality due to different types of environmental stress. I included them to make you aware that they're there, but I'm not going to say much more about them in the course of this presentation. And once we've defined a set of state variables, we can get ourselves a set of equations. If you look, for instance, at the top equation there, I don't know if you could follow my mouse, but the one that I'm winding my mouse around, it says the rate of change of something is equal to some quantity minus something else. All that equation says is the rate of change of what is in one of these boxes is the difference between the rate at which stuff is coming in and the rate at which stuff is going out. And you can do that for all the boxes. For the present purposes, all I think we need to do is recognize that we get a collection of many messy equations. And if you're actually going to use the model rather than get an overview of what it's about, you actually get your hands dirty with these equations. But for now, over the next 20 or 30 minutes, we don't need to get our hands dirty with them. So having given you a very rough overview of what the modeling is about, I actually want to use a small number of examples to let you see how the model can actually deliver useful insights, in particular relating to predictions across levels of organization. And I'm going to present a very, very brief description with no detail of 4 case studies. The first one, which I'll actually summarize in one slide, talks about connections between the subcellular level and the population level using a system with phytoplankton exposed to metal and metal oxide nanoparticles. And I chose that example simply because it's the context in which I've been working, and it's one I'm familiar with. Likewise, there is a second example relating to phytoplankton that this time again that takes us from the individual level to the ecosystem level. Then I'm going to look in more detail at the transition between individual organism response and population dynamics using some work involving the water flea Daphnia, which I mentioned a few minutes ago. And in part to show that we can get a glimpse towards the ecosystem level, and also in part to simply make the point that dynamic energy budget models can also be applied to plants. I'm going to talk about soybeans exposed to again, different types of nanoparticles. One side remark, I've chosen here to use toxic substances, specifically in 3 or 4 cases, those relating to nanoparticles, as my example of environmental stress, because that's one where with which I'm familiar. But the applicability is much broader, and the principles that I'm applying here pretty much carry over into many other forms of environmental stress. As just one example, a colleague, Eric Muller, and I have done some work on ocean acidification using many of the ideas that are similar to what you've just seen here. So let's move on to the first example. And here I'm trying in a somewhat busy slide to summarize the result of a lot of work by a group of about 6 people. The background was that many people have argued and do argue that you can use cellular level experiments on a whole bunch of indicators of a cellular function in order to make predictions about the level of toxic stress that cells are experiencing. And in this project, several people applied these high content and high throughput methods that are applied to marine phytoplankton cells exposed to different types of nanoparticle. In parallel with that, cultures of the same cells getting the same level of exposure were performed in the traditional way, and the rates of exponential growth were measured. Then the paper listed at the bottom gives information about a lot of different correlations between the changes in growth rate and the changes in the various cellular level indicator. Bottom line was the only one that gave a strong correlation was what is called photosynthetic yield, which is a parameter that you can put into a DEB model. The DEB model was essentially simplified to the form of the most elementary differential equation that is ever used in ecology, and the population growth rate involved a parameter called Ke here that was proportional to the photosynthetic efficiency. Thus, the DEB model was capturing the response, but other metrics from high content screening were uncorrelated with the population growth rate. Now, this isn't a great example, except in the sense that if it had failed, it would tell us that there was something really problematic about the approach of following DEB models, because if you change substantially the energy input or a parameter that is known in a known biological way linked to the energy input, and you don't correlate it with the performance of a simple cellular population, something weird would be going on. And thankfully, it wasn't. The next example used a DEB model in a substantially more complicated way, and perhaps more ecologically relevant way, in that it started at the level of the cells and then followed what populations did. The work was performed by a PhD student in UCSB, or a then PhD student, Louise Stevenson. She's the first name in the paper that is cited at the bottom. And the interesting thing that she did was she applied the stressor, in this case citric-coated nanoparticles, but silver nanoparticles, but that doesn't matter. She applied the stressor to batch cultures of a unicellular alga, Chlamydomonas reinhardti, after 1, 6, and 13 days of population growth. The figure at the top left shows you what happened. The green curve is what the batch culture is just what an unstressed batch culture did, with the y-axis being on a log scale. If she introduced with one particular density of stressor, if she introduced them at the start of the experiment, everything died. If she introduced them after 6 days, there was an immediate negative response. Then the culture seemed to hang on for a bit before eventually running into trouble. And when she applied it later, the culture was still hanging in there close to the end of the experiment. The explanation involved studying the interaction of the energy budget dynamics of the algae with changes in the environment. And specifically, it recognized that and this is something you get directly from the DEB model, it recognized that the algae, as they grew, were releasing dissolved organic carbon compounds. The dissolved organic carbon compounds were now able to interact with the stressor, in this case the nanoparticles, and to some extent mitigated their effect and neutralized them. So in order to understand what was going on, we needed to combine a dynamic energy budget model with a model of, in a sense, the biochemical interactions, or the physical-chemical interactions that were going on in the environment. And this indicates, this points to another important feature of the use of dynamic energy budget models, that there are few, if any, ecologically relevant environmental applications where you can use them on their own. They are a tool that characterize what the individual organism is doing, but other stuff is going on in the environment, and there's no way around, nor would we want to ignore that. So as well as putting effort into developing a DEB model for something, it's important to think about interactions with the environment and in the environment. And this example, I think, illustrated it quite nicely. And the next slide perhaps gave some indication as to why the outcome mattered. Louise did some absolutely standard acute toxicity tests where she looked at just what fraction of a set of Daphnia survived for 48 hours when exposed to different concentrations of silver nanoparticles. And if she did it with the medium that was around at the start of the experiment, the red curve showed how the survival decreased with dose. If she did the same experiment with animals at the placed in the water that was around at the end of the experiment, all of them survived. In other words, this two-way feedback where the stressor is impacting the organism, but the organism is impacting the environment that in turn interacts with the stressor is, I would argue, from this kind of evidence, ecologically relevant. The next example goes into more detail about how we use dynamic energy budget models in order to connect the properties of individuals and populations. This is work that was at least what I'm going to present draws very heavily on the work of a PhD or a then PhD student, Ben Martin, who was part of a European CREAM, a project where a large cohort of PhD students was trained in various mechanistic approaches to ecotoxicology. And what Ben's work did was coupled the information you get from dynamic energy budget theory. On the left of that slide is just a reproduction of the cover of Bas Kooijman's book, with well-developed modeling methodology for individual-based population models. And the book on the right is the book by Volker Grimm and Steve Railsback. Steve, as an aside, was a member of one of the Nimbus working groups that lies behind a lot of this work. Volker was one of Ben's PhD advisors. And essentially what the package called DEB-IBM does is it takes the DEB model. I was too lazy to change this figure, so it's now got a yellow background rather than the kind of a bluish background from my previous slide, but it's the same, it's essentially the same characterization. And as I described earlier, lots and lots of these are stuck in a computer, and then the population changes are followed. The programming can be quite tricky in this, but there is a very easy to use free package called NetLogo, which is what was used here, and it is still used by many people. It's not as fast or efficient as some of the more sophisticated individual-based modeling tools that are around, but it is for somebody like me who doesn't do a huge amount, it's absolutely adequate. It's the only one I've ever learned how to use, and it's got great documentation. You just Google NetLogo and you'll get it. So what Ben did was took a large amount of information on the population of dynamics of Daphnia, supplied with food at different rates in a chemostat setup in the lab. He then used these data to parameterize the dynamic energy budget model. He then put the dynamic energy, these parameters into DEB-IBM, and he then predicted, using no new data, what the population would do. And the results are shown on this slide with the bold curve representing the model prediction, the dotted lines around it showing a 95% confidence limits, and the dot showing the data. And you don't need sophisticated statistics to say that see that the model predictions were horrible. In other words, taken just out the box, with actually a very simplistic characterization of mortality, which we hadn't talked about, the model predictions were dreadful. But it turned out to be relatively simple to modify the model. Specifically, we recognized that the weakness was there was a very poor information on mortality, and in particular that poorly fed individuals eventually would experience higher mortality. We'd no idea whether this would impact all stages equivalently, the juveniles more or the adults more, though we suspected that it would be the juveniles more. All this did was involve adding one more parameter to the model. The model could now be run with that parameter tuned, and the best model identified by looking at the goodness of fit at just one food level, and then looking at the model performance at all food levels. And the next slide shows what happened. That one modification to the model took the horrible misfit 2 slides back and gave us a very good fit to the data. And for those who have experience of the challenges of fitting models to real data, especially when you have a large body of data, there's different life stages in these pictures, there's 2 food levels and so on, and there's more in the paper that go along with it. This actually felt quite good, but it still had some limitation in the sense that we did have to tweak the model. We were only doing so well in terms of making predictions. So at this point, what we did was asked, well, one way you can test whether you've got the right mechanisms in a model is to take a completely different data set that wasn't used at all in the model formulation, and can you understand its dynamics? And for this, we turned to a paper written now a decade ago by Ed Macaulay at the University of Calgary, Bill Nelson in Canada, and myself, from experiments done in the Macaulay lab on this time bathtub-sized populations of Daphnia. What Ed and Bill Nelson found was that under identical conditions, they could get two types of behavior. They could get very large amplitude population cycles of the sort that theorists would have predicted using, say, lockable thermodynamics, or they could get relatively small amplitude cycles. And further investigation in some really very clever side experiments that Bill Nelson did showed that the development rate of those experiencing the large amplitude cycles was very fast. That's to say, they become adults pretty quickly. And for those exhibiting the small amplitude cycles, it was much smaller. And in particular, they observed a relationship of these quantities to the cycle period. So what Ben now did was took his DEB-based DEB-IBM model, and this model already includes stochastic variation because not because of anything. We put stochastic variation into this DEB-IBM model. And what you saw there was that as time evolved, spontaneously the system jumped from exhibiting low amplitude cycles to large amplitude cycles back to small amplitude cycles, and there was a change in the maturation time. That's as it moved towards the large amplitude cycles, the individuals developed faster. Obviously, I've described this test far too fast for the details to make sense without reference to the paper. The key thing is that in addition to demonstrating that the DEB-based model could be brought into congruence with data with one adjustable parameter, we demonstrated that it actually was capable of reproducing a pattern seen in completely unrelated experiments. And that provides, I think, an enhanced level of a confidence that the model has some reality embedded in it. And finally, in another paper, Ben looked at the ability of the model to predict the response of parallel experiments in the same system on Daphnia to a contaminant. And what you've got there are a set of 4 panels, or 4 sets of 4 pictures corresponding to different levels of stressor. And all I ask you to see there is that the data points and the model are reasonably matched to each other, thereby demonstrating that the model has applicability. My final example that demonstrates the sort of work that the sort of applicability, and as you'll see in a minute, gives us potentially a small step to thinking at the ecosystem level, involves experiments on soybean in a group at UCSB led by Professor Trish Holden. And her group studied the effects on soybean growth of adding engineered metal oxide nanoparticles to soil, for instance, from atmospheric deposition or whatever source you're thinking of. And the reference for the original paper is at the bottom there. There was, as you see there, 14 co-authors to the paper. There were a huge number of endpoints measured. But the striking result was that with one particular nanoparticle, Syria, the highest toxic response and growth occurred from the lowest concentration of the added nanoparticles. And from the detailed study of the plants, there was evidence of damage to the nodules and the shutdown in the plants of nitrogen fixation. And what was then done in a piece of work that that came out about a year and a half ago, led by Tin Planschek, was that a DEB model, again somewhat simplified from the one you've seen, that did have separate compartments for the above ground and below ground biomass of the plant, for the seeds, and for the rhizobia, the bacteria or bacteroids in the nodules that were actually doing the nitrogen fixation. And that model had various components. We coupled with the DEB model, we coupled what is often called toxicokinetics. That's some equations that, again, describing the interaction with the environment, and in this case, something that led to a toxicant burden in the plant, and that in turn could impact biological production in the plant and also impact the nitrogen fixing microorganisms in the nodules. Oops, something has happened to my picture. Yeah, the model then predicted changes in growth. Something is happening to my display here. I'm just going to pull them all through because apologies for that. The sequencing has gone a little strange. But what you see is that if you now look at the color coding here in the growth equation, that's the control. The lowest exposure gave you the biggest effect on growth. The middle exposure gave you the next biggest, gave you less impact, and the highest exposure gave less impact again. And essentially what the model said was that what was happening was that the N-fixing microorganisms were being killed, and that because nitrogen fixation is an energy-intensive process, very energy demanding, and because these experiments were performed in a greenhouse with a sufficient inorganic nutrient around, that in turn explained the pattern, and indeed Tin's model demonstrated that that was the case. But there was a nice set of experiments that supported this. The model prediction was that nitrogen fixation would vary in a way that was shown here. It is possible to do direct experimental evaluation or characterization of the nitrogen fixation potential of the nodules in at least as proportional changes, and these are shown here. And again, the model was in a broad agreement with the data, and it was again explicitly taking account of bioenergetic processes. I included that example in this lecture because nitrogen fixation is a pretty fundamental process for ecosystem effects. Nitrogen cycling is, of course, a central theme in ecosystem ecology, and this was a way of demonstrating that it too could be impacted in ways described by bioenergetic model. It's also of societal relevance. If a stressor causes even quite small impacts on the growth or yield of a plant, but causes an increase in fertilizer demand, which is what in an agricultural context would happen if N-fixation was switched off. This is obviously problematic and requires attention. So what have we learned from this glimpse at 3 or 4 pieces of DEB modeling? From the first case study, we learned that suborganismal metrics, which are very, very popular, may or may not be predictive of organismal response to stressors. 2 other examples, the phytoplankton study by Louise Stevenson and a population dynamic modeling by Ben Martin showed that ecological feedbacks matter, and both feedbacks via the abiotic environment and via resources matter. Now, to some extent, this is what anyone that does an undergraduate ecology class is told. What's distinctive here, I think, is that the DEB approach gives you ways of quantifying this and modeling it in a predictive way. The third bullet point is, in some ways, a warning that modeling the individual to population connection sometimes does require some data at population level. And that, I think, says something that many ecologists either have experience of or feel in their guts, which is that you can seldom, except in the most trivial situations, actually predict population dynamics from knowledge of what individuals do. But you can go a long way, and you can greatly reduce the amount of population level data that are required. And then the last bullet point says that, our final example, that there is a possibility of some connections to ecosystem processes. I want to make a couple of remarks based on just the first of the first of these, sorry, the first 2 of these bullet points, and how in the 2 Nimbus working groups that were referred to, we've taken account of it. And what I think the early work about suborganismal processes showed was that we ought to be looking at the models, and it's, oh, wait a minute, I've just doubled that slide. That's a better way of putting where I was getting to. Looking at the systems biology models that are out there, because there's a huge amount of effort going into these. Likewise, to look at the feedbacks, we can go to the entire body of ecological theory, half a century's worth. In other words, we can make progress on the issues that have come up really by looking at current and historical literature. I want to say just a little bit more about the connection to systems biology models, and that recognizes that in toxicology, a lot of effort in the past decade has gone into the development and the identification and a study of so-called adverse outcome pathways. And these are the pathways of causal connections that follow what happens when the point where a stressor molecule enters a cell through to the level at the organismal, the level of the whole organism. And this slide, modified from perhaps the seminal paper on these adverse outcome pathways, identifies the macromolecular interactions, the cellular responses, the organism level responses, and the individual responses. And what our Nimbus working group proposed was that we connected these adverse outcome pathway key events, as they call them, with the chemical transformations in the model. And the schematic at the right there is just the schematic that I showed you early on in the lecture. And we can then synthesize everything in, let me just, oops, something has gone kind of crazy with my slide here. I'll see if I can fix it. That's better. We can synthesize that by saying there's a bunch of changes for which we can use adverse outcome pathway or other systems biology models. There is a chain of reactions for which we can use variants and current ecological methodology, but the organism level point, which is the pivot point, has actually is the place where dynamic energy budget models fit in. And really my take-home message from this presentation is that you can think of dynamic energy budget theory as offering some pivot between what I'd call the mainstream of biology, which is concerned with what's at the bottom left of that slide, with the mainstream of ecology. The two Nimbus working groups each produced overview publications that described their work. The Murphy et al. Paper relates to the suborganism to organism work, and the Forbes et al. Paper relates to the organism to ecosystem level response. And that is the take-home message. Thank you.